Hello everyone, welcome back again to my channel. Thank you all for keep loving my videos and keep motivating me. I know obviously you're gonna follow me on Facebook, blog account and Twitter account. Plus I hope you don't forget to like, share and subscribe my channel. Today I'm talking about the gel electrophoresis. I'm talking about what is gel electrophoresis and why we use gel electrophoresis in biotechnology. Now, what is gel electrophoresis? Gel electrophoresis is a technique with separate charged molecule based on its size. And in biotechnology, in most of the case, charged molecule would be DNA or RNA because biotechnology deals more and more with these particles, DNA or RNA. I would like to say molecule rather. And these techniques, gel electrophoresis technique or tool, consist of two different things. First is gel. Gel is a thick, clear, and slightly sticky substance, and we use gel to load our samples and to electrophorese them on certain buffer, certain media, and it also consists electrophoresis. What is electrophoresis? Electrophoresis, in a general term, that describe about the migration and or separation of charged particle or molecules or ions, but it can be separate or migrate under the influence of an electric field. And an electrophoretic system, it consists of two electrodes, either anode or cathode, based on the opposite charged electrodes and connected by a conducting medium which is called electrolyte. Why do we need gel electrophoresis then? I have already mentioned gel electrophoresis help to separate charged molecule based on the size, but why do we need to separate them? Whenever we performing the experiment in lab, sometimes we might contaminate our DNA or RNA. We might have the mixed population of DNA and RNA, and we are looking for some sort of or some desired fragment of DNA and RNA. What can we do in this case is we can do gel electrophoresis and we can separate them and we can identify the right DNA fragment, or we can purify the DNA and RNA based on the gel electrophoresis tool. And another one is we can estimate the size of DNA and RNA fragment. If we have extracted DNA and RNA from some samples or some plants, bacteria, anywhere, but we don't know the DNA and RNA size, then what we can do is we can do the gel electrophoresis and we can compare the size present in the gel with a marker and we can estimate the size of DNA and RNA. And in some cases, we might have the mixed population of protein or contaminated protein. In this case, we want to purify our protein, or in this case, we want to separate the mixed population. So we can do the gel electrophoresis and we can separate them and we can purify them. Well, how gel electrophoresis directly reflect into the laboratory work then? Yes, it reflect either in the visualization form or in conformation form. Well, what happened is when we performing our experimentation in laboratory, um, we might be performing different tasks at the same time. Sometime we might be doing restriction digest, sometime we might doing the extraction of DNA, sometime we might be doing the PCR and some other lot of experiments. And what now after doing the experiments, what we have to do is we have to visualize our result. Mean we have to see like what is the result is. So those to visualize if we are doing in the right direction, if it's the right work, or if we have to visualize what happened during the reaction, what exactly happened during the reaction, then we do gel electrophoresis. We compare the sample band in gel with a marker. You can see the marker band in marker size on the very left hand side of the first picture, and with the reference of the marker, we compare other bands appear in the gel and we visualize how our reactions have gone, how our reactions are going, and how our experimentations are going. And another form comes as confirmation. After doing the experiments, we have some aim, or we wanted to achieve some aim, and we don't know if we are doing the right job. Then after doing the experimentation to confirm what the experiment we have done going in the right direction, or we doing the right job, we do the electrophoresis. Once we do the electrophoresis in the similar way, like comparing with the marker, we confirm we confirm that our work going in the right direction. For example, if we have done the restriction digest of one plasmid, and we wanted to know it has worked properly, what we do the electrophoresis, and we compare that band size with the marker, and we compare it with the theoretical model, and we confirm our experimentation had worked in right way. Now, what is the procedure for running gel? It starts from setting up gel to visualization of the result. And usually in biotechnology, we use two different kind of gels. One is SDS page gel and another one is Agarose gel. Usually in biotechnology, STS PET gel used to separate or denature protein molecules. In some cases, we use STS gel to separate RNA or DNA molecules as well, but generally it used for separating protein molecules. And another one is agar gel, which commonly used for separating DNA and RNA molecules. So I'm starting here from Agaro gel because we have to set gel for Agaro gel, but we don't need to worry to set up gel for STS pay gel because it comes in ready-made way and we can directly use that ready-made gel. So we don't need to worry about the setting up gel for that. So for Agaro gel, what we have to do is we have to wet the right volume of agarose powder, and we have to add the right volume of buffer. Usually we use TAE buffer for agarose gel setting, and usually we use 1:1 weight by volume of agarose and TAE buffer. Once we mix up the buffer and agarose, we have to shake well and we have to heat it up, then we have to let it cool, then we do not have to forget to add stain. If we do not stain our gel, later on during the visualization of the result, we wouldn't see any band. So usually we use cyber safe as a stain in agarose gel, and once we have added stain in our liquid, we have to pour that liquid into a cassette, plastic cassette, which is commonly available in laboratory. Then we do not have to forget like that cassette should be in plain surface. Then another thing we have to take care is to use the right type of comb. The comb we use, like you can see here in the figure, before pouring the gel on the cassette. Um, why we have to use the right type of comb is based on our sample volume and based on our number of the sample. Sometimes we need the deeper well to load the heavy volume of the sample, or sometimes we need like shallow well and we need the larger space of the gel. So based on that, we have to choose either deeper or the very flat kind of comb, and that comb should have the right number of the wells based on our sample number, so that we can maximize our gel surface to load our sample. After that, we pour our liquid into that cassette having the right type of the comb into the plain surface, and we wouldn't disturb it, and we let it there for at least half an hour to 1 hour to set our gel. Then after that, once we set our gel, we have to remove the comb and we have to take that cassette having gel into the apertures. We have to set the apertures in a very right way. In biotechnology lab, we will have gel tank there. So once we remove the comb and once we take the cassette with gel, we have to put that cassette with gel into that gel tank. And we have to take care here as well. We have to pour this gel with TAE buffer. We have to use the same buffer here. So once we fill the gel, we have to provide the power there. So generally towards the well sides, we have to give the negative power, and towards the end in the cassette or in the gel tank, we have to give the positive power. DNA and RNAs are charged particles. They are charged molecules, and they are negatively charged. When they influenced in electric field, because they are negatively charged, they used to move from the negative to positive side. So uh, to migrate them from the negative to positive side, we have to load samples near to the positive electric power. So it is the nature of the DNA and RNA molecules. They move from the negative to positive because they are negatively charged. We have to give power accordingly. So towards the wells where we load the samples, we have to give the negative power. And you are seeing here in the picture like how we commonly set up the gel in laboratory. And next, once after we set up gel or set up apertures, we have to load our samples. The samples could be the PCR product or restriction digest product or colony PCR. What we have to do, we have to stain that product as well. That sample we're going to load, we have to stain. Usually PCR product, they like used to have the green stain, so we don't need to care about that. However, for the rest of other samples, we couldn't see properly like if our sample been loaded, so we used to have stain. Then once we stain our sample, we have to take a small volume of that sample, and we have to load very carefully into the well we have made using our comb. So the sample loading is very crucial thing in gel electrophoresis, because if we haven't load sample in a proper way, it doesn't work. It gives something different result. So we shouldn't have to mix of sample. We have to carefully load sample into the very depth of the well. And therefore we use the very small tips for pipette, and we use the very small volume of the sample. Sometime during the sample loading, our samples used to float away. So it either contaminate the next sample, or sometime like we won't have any sample in that well. So it doesn't give any result, like we won't see anything after that, or we either get different results. So we have to be careful on loading samples. And in some cases, the pipette tip sometimes crack the gel, and once it crack, like the samples used to flow from there. So it it creates the chance to mix up and contaminate the other samples. So loading gel is critical. And after we load the gel, what we do is we run gel. We already, I have mentioned towards the sample loading side, we used to give negative power, and towards the end it would have the positive power. So the particles, molecules, DNA, either DNA or RNA, used to run from negative to positive side. So we have to provide current in this way, and we have to give the right voltage of current. Either it may be 94 voltage or 98 voltage, or sometime it's 100 voltage, based on the experiment you have. And we have to run that sample for certain hours, maybe from half an hour to 1 hour, based on how clear band we are looking for. So if we are running gel for quite bit longer time, sometime it may like migrate totally towards the end of the gel. Maybe it run off towards the gel, and we won't see any band because it had already run off from the gel. So we have to care about that as well. Based on these facts, like taking consideration of these facts, we have to identify the right voltage of current and right amount of the time to run our gel. After we run gel, after the end of the running, we have to visualize the result. Otherwise, like, how can we compare the our experimentation, or how do we know, like, we have this result, or that was the right thing we wanted to do? So we do visualization. And for the visualization, we used to have some softwares and some visualization doc. We usually call the gel doc. And I'm giving here the example of Biotech company, which has the like gel doc, and it has the specific software that connected with the computer that help to visualize our image into the image, like our gel into the image form. So these apertures, this like this kind of setup, useful for visualization of the result. We have to take out the gel from the gel tank, and we pour slightly, press our gel from cassette to like drop into the gel dock, inside the gel dock, and then we close the door and we run the software to visualize our result. And later on in the computer, using that software, and they'll visualize our the apertures visualize our gel, and it take the image, and it gives the result into the image form like this here. So another one is STS gel. SDS gel is a bit different than the Agarose gel, but we do not need to worry about the setting of gel because they're like commonly available in the market in ready-made form. So we have to take care during removing the comb. And once we remove the comb without disturbing the gel, in the similar way like agar gel, we have to load sample in the right well without mixing up samples, and we have to run that gel for certain hour of the time, giving with certain hour of the current. In the similar way here, we separate our molecules. We sometimes use it for RNA and DNA separation, and sometimes we use for protein separation. Usually we use for protein separation. In this is very similar with agar gel, but the only thing, we use the different buffer here, and we use the different gel tank and different kind of gel, because it's a polyacrylamide gel. We don't use agarose. And what happened in the STS pet gel is, like, once we run the gel, if this is the DNA or RNA molecules, after certain hours, we can see the bands after visualizing it under similar kind of apertures like the bio-rad gel doc. And if it's a protein, we also see the similar kind of band in gel you can see in the picture. Only difference between agarose gel and STS pet gel is agarose gel use the agarose and STS use the polyacrylamide, and it generally STS pet gel used to separate or denature protein, and agarose is used for separating DNA and RNA molecules usually. However, the running procedure and visualization procedure is similar. We use the same gel doc to visualize the image, and we use the same software to visualize the image. Only thing we change is our gel type and stain on the software. That's all. Everything is similar. And this much for today. The only thing we have to take care during gel electrophoresis is use the right type of gel, right type of buffer, right amount of the current, and right time of the gel running period. And we have to take care the sample loading. We shouldn't make any crack on the gel, and we have to use the right software to visualize our image. That's all. And pretty much everything similar for the STS pet gel and agarose gel electrophoresis. Only the difference is the gel type. Agarose has agarose and STS has polyacrylamide, and STS generally used to separate proteins, and agarose gel used to separate nucleic acids, DNA or RNA. That's all for today. Thank you everyone for watching my video till end, and if you want me to improve myself, or if you want me to cover something else in next video, please let me inform in the comment section below. um, we will see you in next video. Till then, have a very good day and enjoy your day.